Good morning, everyone. Today, I'm Victoria Nicole and my partner, Materia Zahwa. We're going to review a paper titled Sample Size Calculation for the Differential Expression Analysis for RNA Sequence Data using a Negative Binomial Regression Model. Next. Before researchers perform RNA sequence experiment, they need to answer one important question. How many samples are enough? Why is this important? Because if the sample size is too small, researchers might miss important gains. But if it's too large, it becomes expensive and inefficient. The paper focused on identifying differentially expressed gain, or DIGs, between two groups like healthy versus sick patients or control versus treatment groups. This is useful for diagnosis, predicting disease outcomes, and evaluating treatment response. The challenge is that RNA sequence data are not simple data, they are count data and usually have very high variability, or what statisticians call over-dispersion. Other methods use the Poisson model, but Poisson assumes that the mean equals the variance, and that assumption doesn't work well for RNA sequence data. So the authors propose a better approach using a negative binomial regression model. Next.
Okay, the research never do well without the good methodology. So the data that used in this paper is the RNA sequence gene, which is the type of omics data, which consists of read count of 1,000 genes for two groups. In this paper is control group and treatment group, and the variable that used in this paper is the response variable, which is the RNA sequence itself, and the main predictor is the treatment group indicator, where 0 is the control group and the 1 is the treatment group. And the methodology that used in this paper, of course, the main methodology is negative binomial, because RNA is the count data and over the dispersed data, and then the negative binomial is entered into GLM to compare two groups. Then they calculate the full change and build a sample size formula.
So what did they actually find? The authors introduced two new methods, as if statistical power%, which is considered very good in research studies, but among all methods, the best overall. Why? Because it was stable, accurate, and computationally efficient. Meanwhile, M2 were large, M3 often overestimate sample size, and M4 or M5 were accurate but more computationally expensive. What makes this paper stronger is that the methods were also tested using real breast cancer RNA sequence data, and the results shows that these methods can actually help researchers plan real biological experiments more effectively.
Okay, next. What's actually the strength and the weakness of this study? So first of all, the strength, of course, since they use the negative binomial model in the very over depressed data, so it is more realistic compared to Poisson-based approach. And then the author also compared the new method, which is M1 and M2, which M1 actually computationally efficient and produce sample size that achieves the power that close to desired. And the limitation of the study is only comparing two groups, which is control and treatment group. And then the dispersion parameter is known or fixed for simplicity. And then the paper only rely on simulation study instead of validating the method across many real RNA sequence dataset. Does another approach could work in these problems? Of course. First of all, the methods such as D²2 and H are commonly used because they estimate dispersion and more flexibility. And then the Bayesian approach also works because they could better handle uncertainty in dispersion and estimate, and the machine learning also may help when the data are very high dimensional or involve complex biological pattern. So for the future direction and improvement, we suggest the gene-wise depression, which use gene-specific parameter estimates rather than single common dispersion. And then next, we propose machine learning validation, which is compare and regression sample size estimate with those from the square framework using the cross-validation on the public dataset. And then we suggest multi-group and factorial design to extend formula beyond two groups to handle time courses. And then we also suggest the pilot data uncertainty to incorporate uncertainty in pilot estimate of parameter using bootstrap or Bayesian interval. Yep, I think that's all from us. Thank you for your attention.